Assalamu alaikum, dear students and listeners. We are studying the course of human physiology, and from the course of lectures on blood. Our today's topic is on hemoglobin. What is hemoglobin? Let's discuss about it and try to understand its physiology and chemistry. Chemistry of hemoglobin. Hemoglobin is a red pigment of blood. It's a chromoprotein consisting of two parts. The main part that constitute 96% of the hemoglobin, it's a specific simple protein that is known as globin, also known as histone protein. And the other, relatively, the small portion that constitute just 4% of the hemoglobin, it's a non-specific prosthetic group, also known as iron-containing pigment, which is called heme. Heme is a protoporphyrin compound and consists essentially of four pyrrole groups joined together. As we can see here, it's a protoporphyrin compound and consists essentially of four pyrrole groups that are joined together. If we look upon in detail the structure of heme, we can see here that there are four methyl groups on each corner of the heme group, four vinyl groups, the two vinyl groups and the two propenyl groups. Now here, heme, the porphyrin molecule that can combine with metals forming metalloporphyrin compound. So heme is a metalloporphyrin, where metal is iron, the centered metal. The iron content of hemoglobin is about just 0.34%, and about 3 gram iron is present as hemoglobin in the total amount of blood of an adult. So iron remains in ferrous form, and globin helps heme to keep the iron in ferrous state and to combine loosely and reversibly with molecular oxygen. Here is the complete structure of hemoglobin. The molecular weight of hemoglobin is about 68,000. It contains 4 atoms of iron and 8 atoms of sulfur. The hemoglobin molecule is probably an aggregate of 4 unitary molecules, each having 1 atom of iron and 2 atoms of sulfur, and with a molecular weight of 17,000. The approximate chemical formula is given here. Now the question arises, a very interesting question, that why iron in hemoglobin is ferrous and not ferric? Because the reason is that there are 4 bindings with protoporphyrin ring on the 4 corners, while one binding occurs to the histidine, that is the globin protein. So it means that one free bind to oxygen and or carbon dioxide respectively. Actually, we know that iron in ferrous form contains 6 coordination groups. So from these 6 points, 4 of them bind with protoporphyrin, 1 bind to histidine, that is the main globin protein, and the remaining one is free in order to bind to oxygen or carbon dioxide respectively. While in ferric form, we will have 1 bond missing, that is malfunctional hemoglobin. So heme and globin, they are not isolated. They bind together with the help of iron. Properties of hemoglobin. The most characteristic property of hemoglobin is the easiness with which it combines with oxygen, and the property by which it is dissociated from oxygen. That is the formation of oxyhemoglobin and the dissociation of oxyhemoglobin. So if we take 100, for example, 100 milliliter of water, we can say that 100 milliliter of water will absorb one third just milliliter of oxygen at body temperature under atmospheric pressure. But as compared to water, 100 ml of blood under the same conditions will take up to 20 milliliter of oxygen. We have already seen this amount in the lecture of respiratory system. So we can say that it is due to the presence of hemoglobin in the blood. Blood is capable to saturate more as compared to water. About we can say that about 1200 milliliter of oxygen can be carried by the total amount of blood in an adult man, while 1 gram of hemoglobin combines at normal temperature pressure with 1.34 milliliter of oxygen. So this corresponds to 2 atoms of oxygen for each atom of iron. The resulting compound that is known as oxyhemoglobin, it gives off its full oxygen content when placed in the vacuum. Oxyhemoglobin holds its oxygen loosely, which can be easily displaced by many other gases, forming more stable compounds. For example, with carbon monoxide, NO, nitrogen oxide, and H2S, hydrogen sulfide, they will form carboxyhemoglobin when there is carbon monoxide and hemoglobin, and nitric oxide hemoglobin in the presence of NO hemoglobin and self hemoglobin respectively. Their properties to attach with the hemoglobin varies and depending upon the chemical structure they have. The globin part of hemoglobin directly combines with carbon dioxide and forms carb amino compounds. Actually, we have already seen in the lectures of respiratory system that carbon dioxide travels through blood in the form of carb amino compounds. If we talk about crystallization of hemoglobin, hemoglobin can be easily crystallized. The form of crystals, their solubility, and the easiness of crystallization, that is the forming of crystallization, the crystals, they are characteristic of the species from which hemoglobin is obtained. It means that we can say that the characteristics of hemoglobin from one species varies from the characteristic of the other species hemoglobin. While most bloods, including human blood also, they form rhombic prisms or needle-like crystals. And hemoglobin of different species, they are said to be immunologically distinct. So the distinction lies in the globin part of the molecule and not in the heme part actually. It is known that the amino acid composition of the various globin actually it's the main protein of the hemoglobin. They varies considerably, especially in respect to their cysteine content. So the hemoglobin of different species, they also show different affinity for oxygen. If we talk about isoelectric pH of hemoglobin, the pH, normal average pH ranges between 6.8, while that of oxyhemoglobin, it varies slightly around 6.6. And let's talk about varieties. How many types of hemoglobin exist? There are mainly two types of hemoglobin that exist. That is the fetal hemoglobin and the adult hemoglobin. Fetal hemoglobin differs chemically and even spectroscopically from the adult hemoglobin. It has a greater affinity for oxygen and releases carbon dioxide more readily as compared to the adult hemoglobin. This is due to some difference in the globin fraction. This property helps to compensate the relative anoxia of fetal blood. It means that the babies and the child can take up oxygen very easily, and even in the less amount of oxygen, if less amount of oxygen is available, the babies and the fetus can respire more easily as compared to the adult. At low oxygen pressure, fetal hemoglobin can take up larger volumes of oxygen than adult hemoglobin. And fetal hemoglobin is 70% saturated at 20 millimeter of oxygen pressure, whereas adult hemoglobin is only 20% saturated at this pressure. So A small quantity of fetal hemoglobin persists for some weeks or months after birth, and these characteristics make the child and the newborn easily respire instead of not properly prepared lungs. So the form, if we talk about the forms, the form in which hemoglobin exists in the cell, the actual state in which hemoglobin exists in the red cells is not yet fully understood. The quantity of hemoglobin in the red cells is too great for it to be in simple solution form, and it is also known that it is not present in crystalline form. So from these observations, it has been suggested that hemoglobin remains in some combined form. It is believed that hemoglobin remains absorbed to the lipid material of the stroma and the envelope of red cells. So in certain lower invertebrates, hemoglobin remains free in the plasma. Its inclusion in the red cells has taken place gradually in the course of evolution. If we talk about the causes of inclusion of hemoglobin in the red blood cells, so we can say that though hemoglobin is a protein having its molecular weight very high, that is globin's molecular weight is around 68,000, yet it passes through the endothelial lining of the blood vascular system. So hemoglobinuria is the condition when free hemoglobin is excreted through the urine. If hemoglobin is free in the plasma, then it is excreted through the urine. So that's the reason that hemoglobin does not exist free in the plasma. In plasma, hemoglobin is present normally bound to protein at a concentration of about 5 milligram per 100 ml of whole blood. While in plasma, hemoglobin remains normally in the form of hemoglobin hepatoglobin complex. This complex cannot pass through the urine, and hemoglobin remains binded to this complex. Under normal condition, about 100 to 150 milligram of free hemoglobin is released from red cells due to hemolysis. What is the process of hemolysis? Hemolysis means breakdown of the hemoglobin due to the breakdown of red blood cells. HB or hemoglobin can be bound as hemoglobin hepatoglobin complex by the hepatoglobin present in the plasma. So if released, hemoglobin exceeds the capacity of the hepatoglobin to bind the hemoglobin. Then it is excreted through the urine. It means that if our body produces more hemoglobin than the capacity to bind with the hepatoglobin, so the extra hemoglobin is released through the urine, because extra hemoglobin, the required quantity of hemoglobin bound to the hepatoglobin, but the extra quantity of hemoglobin that remains unbound, it is released through the urine. So we can say that daily about 100 to 150 milligram of free hemoglobin is released from the urine. So it means that this free hemoglobin is produced extra in the extra quantity from the body. The free hemoglobin passes readily through the glomerulus and of the kidney, and of which certain amount is reabsorbed and rest is excreted through the urine. So during reabsorption of hemoglobin through the tubular epithelium, certain amount of it is converted to hemosiderin and excreted in the urine in the form of hemosiderin. So basically we can say that there are two forms of hemoglobin that are released in the urine. Free hemoglobin and the hemosiderin. Free hemoglobin is the form that is not linked to any compound or any complex and remains free. While hemosiderin is the amount of hemoglobin that is reabsorbed during the process of excretion and is converted to hemosiderin, but is extra for the body. So plasma hemoglobin concentration is always associated with the hemosideриnuria through the condition of hemoglobinuria may or may not be happened. Thus, if the hemoglobin was not enclosed by the cell membrane of erythrocytes, it means that if hemoglobin is not bounded to the cell membrane of the erythrocytes, then this hemoglobin would have been passed quickly through the kidney, through the glomerular membrane, and excreted through the urine. Because red blood cell cannot pass through the glomerular membrane in normal conditions. So hemoglobinuria may be caused under the following conditions. So what are the conditions? In strenuous exercise, that is in severe exercise, due to mismatched blood transfusion. It means that in the case of severe exercise, red blood cells are broken down and are used to reproduce energy, and a lot of red blood cells are not available in order to bind with the hemoglobin. So the large quantity of hemoglobin will release in the urine. Similarly, due to mismatched blood transfusion, will degrade and hemolyze the red blood cells. And the black water fever due to virulent type of malaria, and red water fever due to another type of parasite which invades the erythrocyte, causing release of free hemoglobin in the plasma. That is, any infection that cause breakdown of red blood cells will cause the increased quantity of hemoglobinuria. Similarly, paroxysmal nocturnal hemoglobinuria, hypotensity of plasma, thermal or chemical injuries. In transport of carbon dioxide from the tissues to the lungs, the erythrocyte or the red blood cells with its hemoglobin plays an important part because the enzyme carbonic anhydrase is present within the erythrocytes. So these red blood cells or erythrocytes help to bind carbon dioxide and help in the transportation of carbon dioxide from tissues towards lungs. If hemoglobin, instead of staying within the capsules, remains free in the plasma, as occurs in the case of hemolysis, many many injurious effects, that is, the dangerous effects will be produced. So the degree of the deleterious effects or these dangerous effects will depend upon the number of red blood cells that are broken down and the rapidity of the hemolysis, the process of hemolysis. Briefly, if we discuss the the bad effects or the disadvantages that will be produced, it means that viscosity of blood will rise as a result of hemolysis. The colloidal osmotic pressure, normally about 30, will rise to 100 millimeter of mercury or even more. This will seriously disturb the interchange of various substances and nutrients in the capillary area, and will also disturb the formation of urine. Similarly, loss of hemoglobin will reduce the amount of blood buffers, because we know that blood acts as buffer, and will cause acidosis in the body. Similarly, loss of hemoglobin will reduce the oxygen carrying capacity of blood, thus producing anoxia and acidosis. Bile pigments will be produced in larger amounts by the rough endoplasmic cells from the released hemoglobin, and in this way, additional pressure will be put upon the liver to deal with them, with the bile pigment. While being excreted through kidney, hemoglobin will be precipitated in the acid urine, and in this way, the kidney tubules will be blocked. This will cause serious disturbance to the kidney function. Let's talk about normal hemoglobin count in an average adult and child. Average hemoglobin content in blood is 14 to 16 gram per deciliter. We can say that 16 is for male and 14.5 is for female. So the HB value depends upon the age and sex of the individual. If we consider age, then at birth, there is almost 25 gram per deciliter quantity of hemoglobin is present in the blood. While after 3 months, the quantity falls down to 20 gram per deciliter. And after 1 year, it falls to 17 gram per deciliter. While from puberty onward, 14 to 16 value is maintained. At the time of birth, hemoglobin count is high due to the increased number of red blood cells and the increased supply need of oxygen supply to the blood to the body. Let's talk about hemoglobin functions. The main functions, the major functions of hemoglobin, they are transport of gases, that is transport of oxygen and carbon dioxide, and the buffer action. When oxygen binds with hemoglobin, a physical process which is called oxygenation occurs and results in the formation of oxyhemoglobin. The iron remains in ferrous state in this compound, that is in the oxyhemoglobin. Oxyhemoglobin is an unstable compound and the process is reversible. That is, oxyhemoglobin is readily converted to hemoglobin and oxygen is released towards the tissues. When oxygen is released from oxyhemoglobin, it is reduced. It's called reduced hemoglobin or ferrohemoglobin and is be ready to take another oxygen molecule. While if we talk about transport of carbon dioxide, that is travel of carbon dioxide from tissues to lungs, carb hemoglobin is formed when carbon dioxide is combined with hemoglobin. So it is also unstable compound and the combination is reversible. That is carboxyhemoglobin is carbaminohemoglobin is readily converted to carbon dioxide and carbon dioxide is exhaled out easily. The affinity of hemoglobin for carbon dioxide is 20 times more than that for oxygen. And it's a very interesting fact. If we talk about buffer action, hemoglobin acts as a buffer and plays an important role in the maintenance of acid-base balance. We have already seen this phenomena in the lecture of respiratory system. Let's discuss about heme synthesis. Heme, what is heme? As we have already seen in the in our last slide, that here it's the structure of heme. It's a combination of protoporphyrin ring and the iron, that is the metal compound. So heme is a combination of protoporphyrin plus iron. And what is protoporphyrin? It's iron free heme, obviously. Protoporphyrin consists of four pyrrole rings on the four sides. While we have already discussed that iron have six coordination number, four are bound with four pyrrole rings of protoporphyrin. One is bound with histidine, that is an amino acid, and one remaining is free to bind with oxygen or carbon dioxide as the need may be. So if we talk about synthesis of hemoglobin, hemoglobin is synthesized inside the red cells in the bone marrow. A number of factors are necessary for the synthesis of hemoglobin. They are as follows. That is, the first class proteins. First of all, they are important. It is necessary for the synthesis of globin part of hemoglobin. Certain individual amino acids such as histidine and phenylalanine, leucine, etc., they have been found to possess a special stimulating action on hemoglobin formation. While a diet that containing kidney, spleen, heart, and certain fruits, they are very helpful in order to prepare this protein. Four kinds of globin peptide chains are present or take part in the formation of heme group. Alpha chain, beta chain, gamma chain, and delta chain. They have been isolated from heme group. Human hemoglobin contains two alpha and two beta chains, and heme portions are attached to the alpha and beta chains. So in the synthesis of hemoglobin, metals are also very important. If we talk about metals, we know that it contains iron. So we can say that iron is an essential constituent of hemoglobin. And daily intake of almost 12 milligram of iron is sufficient for an adult body. While apart from iron, there are it's also needed copper, manganese, and cobalt. These metals, particularly copper, help in the incorporation of iron in the protoporphyrin molecules for the formation of metalloporphyrin. So the ratio between copper and iron in the daily diet should be 1 to 100. Endocrine, of the endocrines, only thyroxine is of proved value in order to synthesize the hemoglobin. And out of vitamins, vitamin C and B12, they are definitely helpful in this respect. It is also believed that folic acid, riboflavin, nicotinic acid, and pantothenic acid and pyridoxine also play some part in the formation of hemoglobin. So let's talk about porphyrins. First of all, if we look into the steps of the synthesis of hemoglobin, it actually starts in the pro-erythroblastic stage. However, hemoglobin appears in the intermediate neuroblastic stage also. Production of hemoglobin continues until the stage of reticulocyte. Heme portion of hemoglobin is synthesized in mitochondria and the globin part is synthesized in ribosomes. We can also see this phenomena in the diagram, that heme portion of hemoglobin is synthesized in mitochondria and globin part is synthesized in ribosomes. As we know that globin part is a protein part and proteins are synthesized on the ribosomes. Heme is synthesized from succinyl-CoA and the glycine, the simple molecules, two simple molecules, succinyl-CoA that might come from the Krebs cycle and the glycine, a simple amino acid. So in the first step, which occurs in mitochondria, that is the formation of heme group. Two molecules of succinyl-CoA combines with two molecules of glycine and condense to form 5-amino levulic acid, ALA, by the help of ALA synthase enzyme. Here, ALA the product 5-amino levulic acid is transported to the cytoplasm. Two molecules of it combines to form profobilinogen in the presence of ALA dehydratase. Let's look upon this phenomena. Here in the mitochondria, the first step takes place in the mitochondria, where two molecules of succinyl-CoA and two molecules of glycine combine to form, in the presence of ALA synthase, combines to form to produce amino levulinic acid. This amino linoleic acid, now it is transported to cytosol or the cytoplasm. In the presence of next enzyme, ALA dehydratase, it is converted to PBG, porphobilinogen compound. Let's go back. Now, porphobilinogen is converted into uroporphobilinogen 1 by the help of uroporphobilinogen synthase. We can see here the process, the formation of porphyrinogen 1 with the help of HMB synthase enzyme. Then uroporphobilinogen 1 is converted into uroporphobilinogen 3 by the help of another third synthase enzyme. From uroporphobilinogen 3, a ring structure of coproporphyrinogen 3 is formed by the help of a decarboxylase enzyme. In the next step, coproporphyrinogen 3 is transported back to the mitochondria, where it is oxidized to form protoporphyrinogen 9 by coproporphyrinogen oxidase. We can see here the phenomena, the reaction step, that the coproporphyrinogen 3 is transported back towards mitochondria and with the help of enzyme oxidase enzyme. In the next step, protoporphyrinogen 9 is converted into protoporphyrin by protoporphyrinogen oxidase. And then this protoporphyrin combines with iron to form heme in the presence of ferrochelatase. Whole processes, this is the diagrammatic representation of all steps of synthesis of hemoglobin. So then starts the formation of globin that takes place in the ribosomes. Polypeptide chain of globin are produced in the ribosomes. Four types of polypeptide chains that name alpha, beta, gamma, and delta chains are produced on the ribosomes. Each globin molecule is formed by the combination of two pairs of chains, and each chain is made of 141 to 146 amino acids. That is, two alpha chains and two beta chains are present in the human hemoglobin. Adult hemoglobin contains two alpha chain and two beta chains, and the fetal hemoglobin contains two alpha chains and two gamma chains. Each polypeptide chain combines with one heme group molecule. Thus, after the complete configuration, each hemoglobin molecule contains four polypeptide chains and four heme molecules. Let's talk about the amount of hemoglobin in normal blood. The average adult figure, irrespective of the sex, we know that it contains 14 to 16. Our next, our next slide is blood groups. What are blood groups? There are four main blood groups, as we already know about it. That is, there are four types of blood. Blood group A, B, AB, and O. And blood group is actually determined by the genes that we inherit from our parents. Each group can be either divided into two RH factors, RH1, RH positive and RH negative, which means that there are total 8 blood groups. That is, we can say that we have total A positive, A negative, B positive, B negative, AB positive, AB negative, and similarly O positive and O negative. That is 8 blood groups. Apart from RH factor, there are also some other factors like M factor, N factor, but they are very less found in the population, in human population. Some other blood group characteristics, except the above three, they are also present, but their certainty of presence is very less as compared to the ABO. So the main blood group, that is ABO blood group. Hello. What are antibodies and antigens? Blood is made up of red blood cells, white blood cells, and platelets in a liquid called plasma. The blood group is identified by the antibodies and antigens present in the blood. Antibodies are proteins that are found in plasma. They are part of our body's natural defense system, and they recognize foreign substances such as germs, bacteria, and infections that attack our body and alert our immune system, which destroys these infections and germs. Antigens are protein molecules which are found on the surface of the red blood cells. Now the question arises, what is the importance of antigens and antibodies? The red cells of any individual, they contain antigens on their surface that correspond to their blood group and antibody in the serum. So we can say that antigens are present on the cell surface and antibodies are present in the serum that identify and combine with the antigen sites on the surface of red blood cells, so of another type. It means that whole process occurs on the surface of red blood cells, and the reaction between red cells and the antibodies usually result in formation of clumping, and the process is known as agglutination of the red cells. Therefore, the antigens on the surface of these red cells, they are often referred to as agglutinogens. Antibodies are also part of the plasma protein and are known as immunoglobulins. That is, they are globulins, they are globulin proteins that take part in the immune system. So with the reference to this function, they are known as immunoglobulins and are classified by their molecular size and molecular weight that they possess, and by several other biochemical properties. Like most blood group antibodies, they are found either on immunoglobulin G, so we call it IgG, or immunoglobulin M molecule, and we call it IgM. Basically, these two types, they are majorly found in the antibodies IgG and IgM. But occasionally, the immunoglobulin A, that is IgA class, may also exhibit some blood group specificity. While naturally occurring antibodies, they are result of immunization by substance in nature that have structure similar to human blood group. These antibodies, they are present in any individual despite the fact that there has been no previous exposure to the corresponding red cell antigens. For example, if we call that for example, antigen A in the plasma of a people of blood group B and antigen B in the plasma of people of blood group A, they are present. What is the ABO blood group? Blood system. There are, we have already discussed that there are four main types of blood groups. Blood group A that has A antigen on the red blood cell and anti-B antibodies in the plasma. That is, antigens, type of antigens that blood group A contains, it's A antigen and B antibody. While in blood group B, that is, they are opposite to each other. While in blood group B, it contains B antigen and with A antibody in the plasma will be present. So if someone that has a blood group A, it has a A antigen and B antibody. But if it will be given mistakenly blood group B from any donor, then blood group B will contain B antigen and A antibody. So A antibody from the donor blood group will fight and combat against the A antigen of the recipient blood group, and they will destroy the red blood cells. And due to hemolysis, the patient may die. While in blood group O, it has no antigen, but both anti-A and anti-B antibodies are present in the plasma. So it means that because this blood group O person does not contain any of the antigens, neither A, neither B. So it can be, this blood can be given to blood group A and blood group B. While if we talk about the fourth type, blood group AB, which has both antigens, blood antigen A and antigen B, but it has no antibody. So it means that because of presence of both antigens A and B, this blood group cannot be given to blood group O, but it can take blood, this person can take blood from a donor of blood group O as it has no any antibody present. Because there is no antibody, so obviously this blood group person or the recipient blood will not fight against the blood group O, which does not have any antigen. Blood group O is the most common blood group. Almost half of the UK population, that is 48%, it has blood group O. But keep in mind that there are two, considering the RH factor, there are two blood group O positive and blood group O negative. Blood group O positive is frequently seen, but blood group O negative, on contrast, is very less found in any population. Receiving blood from the wrong ABO group can be life-threatening. For example, if someone, as we have already discussed, that if someone with group B blood is given blood group A, their anti-A bodies will attack the group A cells. This is why group A blood must never be given to someone who has group B blood and vice versa. But as group O red blood cells do not have any A or B antigen, it can safely be given to any other group. There is another system. Okay, a group A, if we call about antigen group A or blood group A, it is subdivided into group A1 and A2. And similarly, group AB is also subdivided into two groups, A1B and A2B. Approximately, it is seen that approximately 80% of group A belongs to the subgroup A1 and 60% of group AB belongs to the subgroup A1B. While on contrast, group A2 is very less found in the population. and the remainder belong to the subgroup A2 and A2B, but they are very less in the percentage. While if we talk about blood group B, there is only B that exists. There is no B1 and B2. The anyone of the patient or the human that can occupy a given position on the chromosome. If we talk about ABO blood group, the O gene is assumed to be recessive gene, and consequently, the genotype AO and BO they are expressed as A and B respectively because O is the recessive phenotype and A and B both they are dominant phenotypes. So the finding of subgroup A1 and A2 suggests that there are four alleles in total, A1, A2, B, and O, leading to six phenotypes in total. It means that if any person has a phenotype O, its genotype would be obviously O. But if any person that has a phenotype A1, it would show, it might have genotypes A1, A1, A1O, A1A2. While in case of phenotype B, there is only possibility of having genotype BO and BB just. Let's talk about RH factor. What is RH system? RH agglutinogens. There are 6 or 3 pairs of RH agglutinogens, C, D, E. C, D, and E, they are Mendelian domains, while small c, D, and E, they are recessive domains. The red blood cells actually sometimes have another antigen, a protein that is known as Rh antigen. If this is present, the blood group is Rs positive. And if it's absent, the blood group would be Rh negative. This means that we can find 8 total blood groups. About 85% of the UK population is Rh positive, while out of this 36% of the population has O positive, the most common type. In most cases, O Rh negative blood, that is O negative blood, can safely be given to anyone. It's often used in medical emergencies when blood type is not immediately known. Because blood group O negative does not contain any Rh factor and even no antigen is present in this blood group. Blood group test. To work out on the blood group, how blood group can be determined. Actually, in this process, in the test, red blood cells, they are mixed with different antibody solutions. And if, for example, the solution contains anti-B antibodies and you have B antigen on your cell, it means that your blood group is B. It will clump together. And if the blood does not react to any of the anti-A or anti-B antibodies, it's blood group O. A series of tests with different types of antibody can be used to identify the blood groups. The pregnant women are always given a blood group test. This is because if the mother is, it's very important to know that if the mother is Rh negative or Rh positive. For example, if a mother is Rh negative, but the child has inherited Rh positive blood from the father, it could cause complications if it's left untreated. And if it left untreated, normally the newborn child may die before the birth. Rh negative women of childbearing age should always only receive Rh negative blood. Chemistry of the blood group. What are substances? The exact chemical structure of some blood groups has already been identified as they have gene products, that is, those molecules synthesized as a result of an inherited genetic code on a gene of a chromosome and assist in synthesizing the antigens on the red blood cell surface and determine the blood type. Blood group antigens, they are present on glycolipid and glycoprotein molecule of the red cell membrane. The carbohydrate chains of the membrane glycolipids, they are present towards the external surface of the red cell surface and carry antigens of the ABO blood group. H, L, H system, I, and P system also. They carry genes for these systems if any RH system or RI or P system is present. Glycoproteins, which transverse the red cell surface, have a polypeptide backbone to which carbohydrates are attached. What is the method of identification of blood group? The basic technique in identification of the antigens and antibodies of blood group, it's the agglutination test. Agglutination of red blood cells result from antibody cross linkages established when different specific combining sites of one antibody react with antigen on two different red blood cells. By mixing red cells, that is their antigens, and serum, that is their antibody, either the type of antigen or the type of antibody can be determined depending on whether a cell of known antigen composition or a serum with known antibody specificity is used. So in this way, this is the basic principle of agglutination test. That is, by using a solution of known antigen composition and known antibody composition, we can find out the blood group, exactly the blood group that a patient has. In its simplest form, we can say that a volume of a serum that contain antibody is added to a thin suspension of red blood cells, which are suspended in a saline solution in a small tube with a very narrow diameter. And this tube will then be incubated at the appropriate temperature, normally 37 degree. The red blood cells will have settled to the bottom of the tube because of their weight and the molecular size. These sedimented red cells, they are examined macroscopically, that is with the naked eye for agglutination, or they may be spread on a slide and can be viewed through a low power microscope. An antibody that agglutinates red cells when they are suspended in a saline solution is called a complete antibody. With powerful complete antibodies such as antibody A and antibody B, we can easily placed on a slide and together with a drop that contain a red cells in it in a suspension. After stirring, the slide is rocked and agglutination is visible in a few seconds or few minutes, up to few minutes. It also is always necessary in a blood grouping to include a positive and a negative control for each test. Let's talk about blood transfusion. What is a blood transfusion? Blood transfusion is a routine medical procedure in which donated blood is provided to, uh, to you through a narrow tube placed within a vein in the arm. This potentially life-saving procedure can help replace blood loss due to surgery or injury. A blood transfusion also can help if an illness prevents the body from making blood or some of the blood components correctly. Blood transfusions usually occur without complications. When complications do occur, they are typically mild. People receive blood transfusions for many reasons, such as surgery, injury, disease, and bleeding disorder. Actually, we know that blood has several components, including red cells, which carry oxygen and help remove waste products, white cells that help our body to fight against infections, and the plasma in the liquid part of the blood, and the platelets that help our blood to clot properly. A transfusion provides the part or parts of blood that we need. With red blood cells being the most commonly transfused, you can also receive whole blood, which contains all the parts, that is, the white blood cell, red blood cell, platelets, etc. But whole blood transfusions are not much common. The commonly they are erythrocyte transfusion, that is red blood cells transfusions. What is the risk that can occur to the blood transfusion? Blood transfusions are generally considered safe, but there is some risk of complications also that exist. Mild complications and rarely severe one can occur during the transfusion or several days or more after. More common reactions include allergic reactions, which might cause hives and itching and even fever. There are some blood-borne infections. Blood banks screen donors and test donated blood to reduce the risk of transfusion-related infection and to reduce the risk of infection such as any HIV or hepatitis B or C infection. But the infections, they are very rare. Other serious reactions may include acute immune hemolytic reaction or delayed hemolytic reaction or graft versus host disease. This table shows the blood type compatibility. That is, if someone has a blood group A positive, it would be given A positive or AB positive, and it might receive, it can give blood to just A positive and AB positive person, but it can receive blood from A positive, A negative, O positive, and O negative person. We can notice here that O positive and O negative person, we can find here in the blood type A, that is, they can donate blood to blood type A, blood type O, B plus, and AB positive. While in case of A negative and O negative, it can donate just blood to A positive, A negative, O negative, B negative, and AB negative patients. So the table demonstrates the donation and the person that can donate blood to a specific blood group and can receive blood from the specific blood group. We have already talked about the risk and some blood-borne infections. So our today's lecture related to physiology of the blood and the hemoglobin composition, their structure, physiology, and the synthesis ends here. Thank you.